Hi everyone, in this video I'd like to talk about carbohydrates. So for our introduction to carbohydrates, let's first off talk about the main functions that carbohydrates possess. And here on our planet, and that's chemistry we'll be worrying about in this class, carbohydrates serve as energy storage molecules. So you're probably all familiar with the following equation that I've shown here. Carbon dioxide can be added to water in the presence of sunlight. So here's our little sun, okay, irradiating this, and plants can then fix carbon dioxide and water, make carbohydrates, and then a byproduct is oxygen gas. You're probably also familiar with the opposite of this. If you take carbohydrates, burn them in the presence of oxygen, the byproducts are CO2 and water. Okay, so the top one is called photosynthesis. Okay, so plants do that. The bottom reaction, the burning of carbohydrates, is called oxidative metabolism. Okay, and all other organisms do this. We use carbohydrates. But ultimately, the energy source for all of us is the sun. This represents what's called the carbon cycle. You might have heard that phrase in maybe your intro bio classes or an ecology class. So carbohydrates serve a critical feature or function here on our planet. They serve as energy storage molecules. Second, sugars or carbohydrates provide carbon atoms for biosynthesis. And what I mean by that is that our bodies and all other organisms need to be able to use the carbons found in carbohydrates to synthesize things like proteins, lipids, DNA, and RNA. So those carbons to build these molecular types of molecules are ultimately coming from, well, really the carbon dioxide that's fixed in photosynthesis, but then we have various mechanisms in our body to then convert these carbohydrates into these sorts of things. And then lastly, carbohydrates can serve as recognition molecules. Okay, so when I say recognition molecules, what I mean is that oftentimes cells will have coatings of different types of sugars that allow our bodies to sort of identify which organs maybe cells belong to, or if in fact we have an invading cell. So the best example of this is the ABO blood type. And this ABO blood type is defined by the specific carbohydrates that are then coating our red blood cells. So that covers the functions of carbohydrates. In the next section, we're going to take a look at the actual chemical structure. So let's take a look at the classification of carbohydrates. Carbohydrates can typically be grouped in two main groups of carbohydrate, the aldose and ketose sugars. This first sugar I've drawn out here, notice it's got 1, 2, 3, 4, 5, 6 carbons, and carbon number 1 is in fact an aldehyde. This is actually the molecule called glucose. But we could classify this as an aldose sugar, so it contains an aldehyde functional group. Okay, so this entire sugar could be called an aldose. It also can be classified based on the number of carbons found in the sugar. So glucose happens to have 6. There are others that have 5, some that have 4, and so on. So because this has 6, this could be called a hexose sugar. And specifically, glucose would be an example of an aldohexose. So we're actually combining both the aldehyde classification and the number of carbon classification in this aldohexose here. So we'll learn a lot more about glucose as we go throughout the semester. This is probably a molecule you want to memorize the structure of. Next, we have a compound over here that instead of having an aldehyde, has a different functional group. This is a ketone. Okay, and as you might expect, this is going to be called a ketose. and there are 1, 2, 3, 4, 5, 6 carbons, so this would be called a hexose as well. This particular one is specifically called fructose. You might have heard of high fructose corn syrup. Well, there's the structure of fructose. But fructose could also be classified as a ketohexose. So again, carbohydrates come in sort of two main flavors, if you will. They come in the aldose category, contain aldehydes, which are they're always labeled with carbon number 1. And then ketose sugars, which contain a ketone. Most of the times, the ketone occurs at carbon 2 of the sugar. There are some examples where that's different. But glucose in particular, this is a structure that you'll definitely want to know as we proceed forward. All right, the last thing I want to talk about in this brief introduction to carbohydrates and their structure is to talk about a different way of drawing carbohydrates. So here I have, again, the structure of glucose. Remember, it's an aldose. It's got 6 carbons. And I've drawn it such that the OH groups actually are in a particular arrangement. And it turns out that for glucose, it does actually matter which side of this chain of carbons you put the OHs on. So the structure that I've drawn would be sort of the expanded form where I'm showing all the carbons and hydrogens and their bonds. If we were to draw the three-dimensional structure of this, okay, what would be happening here is we're going to envision all of the vertical bonds sort of coming out towards me. So let me redraw this in this sort of three-dimensional way. I'm just going to draw my chain of carbons first, and then I'm going to put in wedge bonds here, and wedge bonds over here. And then technically I need to make each of these sort of a dashed line going back this way. Okay, so now if I fill in my OHs, this would be a more true representation of the three-dimensional structure of glucose. However, you can see that took me quite a while to draw that out. This would be the three-dimensional representation of glucose. So a much easier way was proposed back in the late 1800s by a very famous chemist. His name was Emil Fischer. And the type of projection or drawing that he devised is named after him. It's called the Fischer Projection. And essentially what we're going to do, we're going to make it so that all of these lines are essentially flat. So let me draw the Fischer projection of glucose over here. So I'm just going to draw a straight line down to CH2OH. And then for each of these positions where I have a carbon, I'm just going to draw a line. Like that. Okay, and I'm going to place the OH groups on the correct side of this line. So for glucose, all of the OHs are on the right-hand side except this one down at carbon number 3. It flips over there. These guys are here, and then I'll fill in the rest of my H's. So in essence, each place where a line crosses represents a carbon with its associated four bonds. But in the Fischer projection, the horizontal lines would be going back into the page towards or away from you. So horizontal is back, and the vertical lines are out towards you. So in the Fischer projection, just by convention, that's what's thought. So instead of having to draw all of these dashes and wedge bonds, we'll just draw it like this, much simpler to just draw a straight line. But we do recognize that each of these does have three-dimensionality to it. So the molecule glucose actually has 4 chiral centers, carbon 2, 3, 4, and 5. We could potentially draw different mirror images, isomers, either enantiomers or diastereomers. But this is the Fischer projection. Okay. One last thing while I have this up here when we're talking about Fischer projections. We can classify sugars based on the orientation of the farthest chiral carbon away from the carbonyl. So here I have my aldehyde carbonyl. If I go to the farthest chiral carbon away from that, that would be carbon number 5. If if I look at the position of this OH relative to the vertical line, you'll notice it's on the right-hand side. So because it's on the right-hand side, this is called a D sugar for dextro. Dextro meaning right-handed. So this is actually called D-glucose. If this OH at carbon 5 were actually on the left-hand side, and it was essentially the mirror image of all the other positions, the enantiomer of D-glucose would be called L-glucose. So in general, when you're looking at Fischer projections, you can classify them as either D or L sugars by looking at, again, the farthest chiral carbon away from the carbonyl carbon, and then identifying in the Fischer projection if the OH group is on the left or right of that bond.